The deep biological problem that we started with was trying to understand how cells change their shape during development and what are the forces involved with those cell shape changes.
In my PhD, I've been working a lot about how living things sense mechanical force. I was also interested in how they sense gravity. In science, we spend a lot of time using the tools that we have to ask interesting questions and go about trying to answer them. And this is also a moment where you get to step back a little bit and say, can we build a new tool that will let us ask things that nobody else can ask? Being able to build a new device that could unlock what was invisible in the biology world so that you can just ask new questions. And that's particularly a great thing about collaborating at Carnegie Mellon, because you get people who are working together to build tools. And that's just a really exciting part of science for me.
At the time, there was a scientist named Manuel Farge who discovered that if you squeeze or poke embryos that it would turn certain genes on. But he was doing that at one embryo at a time. So what I did was turn to Phil LeDuc in his lab and said, can we do this with hundreds of embryos at a time so that we can look at a larger scale at the omics, the proteomics and transcriptomics, to see what's happening when you mechanically compress an embryo.
One of the amazing things there is mechanics is all over the place in your body, right? So your blood flows, your heart pumps, your bone, your muscle, even concussions are all related to mechanics, and that mechanics affects your biology. And so we were looking at this in this project in terms of Drosophila and organism level mechanics and how it affected development of these organisms. And so our hope is that this study will help us be able to address and treat diseases of mechanics types in humans in the future.
One thing that might be confusing is, why do we care so much about fruit flies? Do we really want to solve diseases in fruit flies? But we can learn a lot about humans, turns out, from really small organisms. Fruit flies are a model organism that we've been working with for over 100 years. It's really well mapped out. It's really powerful what we can do with fruit flies. So to me, the fact that we're doing this in fruit flies is one of the most exciting things scientifically, because it means you can take all of these mechanical questions that people used to be looking just at cells or just at cell groups, and now take it to a whole organism, and that organism is the fruit fly. So that's a really, really exciting part about this for me, that we're able to bring all of these questions and all of these techniques for the first time to hundreds of fruit flies.
So this is the device that we have developed, and it looks like a really simple piece of polymer here, but actually it has some micro features in it which has been fabricated and built in the clean rooms of the Carnegie Mellon University, and it has many tiny long channels surrounded by very tiny deformable walls which are tinier than your hair actually. And when we put the embryos inside the channel, these tiny walls that are even narrower than your hair can deform in a very precise way, and we can measure and adjust this deformation at a micron level sensitivity in such a way that we can apply these mechanical forces to the embryos in a very sensitive manner. So it looks very simple and not fancy, but the thing that it is doing is actually the state of art.
The great thing is this is Carnegie Mellon, and this is the exact kind of thing that they support, which is very different than at a lot of places. And so it's a great thing about Carnegie is that they actually will support you on these new projects where you're trying to basically develop a new line of research, and that's a very unique quality and characteristic of Carnegie Mellon.
And what it means is we can now ask questions that we've never been able to ask before about how living things sense mechanical force, which could teach us a lot about mechanic-related diseases and about what happens when people are deprived of mechanics, like in space.
So the beauty of this type of interaction between the LeDuc lab and the Minden lab is that we're now getting students from my lab learning engineering tools and students from Phil's lab learning biological tools, and so that will create the future sort of biomedical scientists who can merge different technologies and sort of be truly interdisciplinary in their research.
It's always interesting. I say If you have two faculty members who have a great idea and they sit there and talk about it, that's great. But unless you have someone to actually do the work, it's just two faculty members talking to each other. And so Arden and Utku are fantastic. They were team. One is a biologist, one is very much an engineer. They worked together, they developed this new direction, and they got it done in a tremendous way.
When you get interesting teams of people who don't usually talk to each other, who have different experiences and different expertise, and you get them to come together in a productive way where they're working together on a goal, you can get really exciting stuff.